HIV, Reality or Artefact? By Stefan Lanka. An error can never become true, however many times you repeat it. The truth can never be wrong, even if no one ever hears about it. Mahatma Gandhi. For the past 10 years or so it has been the accepted wisdom that the human immunodeficiency virus, HIV, causes AIDS. It supposedly occurs in many body fluids, and its transmission, especially in semen and blood to a new host, triggers a slow but inexorable progression to AIDS and ultimately death. To infect another cell, HIV must at some stage in its life cycle exist as a separate and identifiable entity. What has been ignored and kept from public awareness is that there has never been a workable HIV test and that the definition of positive has always changed according to the views of different organizations dealing with it, changed also according to the kind of tests used and changed from laboratory to laboratory performing the tests. Its techniques have not been standardized, and the magnitude and consequences of interlaboratory variations have not been measured. Its results require interpretation, and the criteria for this interpretation vary, not only from laboratory to laboratory, but also from month to month. 1. The dispute over who discovered HIV. 2. Was a distraction from the question of whether the virus actually exists at all. The public was impressed that if a president and a prime minister 3 had to meet to resolve attribution, then the thing they were negotiating about must be real. In 1993 a research group from Perth, Australia, succeeded in publishing a paper on the HIV test. 4. Since then anybody could have read for him or herself that no AIDS test could ever work, because HIV has never been isolated, nor even shown to exist. Since AIDS research and the media have largely ignored any critique of HIV equals AIDS, especially the essential question of whether HIV really does exist, it is time to call again for a reappraisal of the whole HIV/AIDS hypothesis. In going back to the origins of HIV virology and telling the HIV story, a view will be presented which will make clear that HIV itself, the very object of this Manhattan Project of modern medicine, AIDS research, does not exist. 5. A little virology. Viruses are essentially just packages of genetic information enclosed in a coat which consists of proteins. They can reproduce themselves only by infecting a suitable host cell and appropriating the chemical machinery they find there. The proteins making up the viruses are characteristic for each species of virus. Apart from enveloping and transporting the genetic information intact, the composition of proteins for a given virus results in a specific shape for the virus particle. This much is generally known. Less well known is the existence of other particles which look like viruses but aren't, and are nonchalantly referred to as virus-like particles. Such particles are far from rare, found, for example, always in placentas, and very frequently in the artificial environment of laboratory cell cultures. They have served to muddy the waters considerably as far as AIDS research is concerned, because particles just like these have been called HIV to date. None of these has been characterized and shown to exist as an entity which one may justifiably call a virus. One root of the belief in the AIDS virus. In classical theory, DNA encodes the genetic material of heredity, which is then transcribed into messenger RNA, which in turn specifies the assembly of amino acids to construct the proteins of all living beings. In 1970 an enzyme, biological catalyst, was discovered in extracts of certain cells which was capable of converting a molecule of RNA into DNA. This was a revolutionary discovery, because it overturned a fundamental tenet of molecular genetics, namely, that the flow of information was strictly one way and never reversed. It had hitherto always been thought that DNA was transcribed, converted into messenger RNA, and that the reverse process from RNA to DNA was impossible. The enzyme responsible became known as reverse transcriptase. 6. And a lot of new myths arose. An era of the past. Cancer caused by viruses. It was believed that the new enzyme was a marker for a virus, because the cells in which it was detected, and which were used to study cancer, 7, were thought to have become cancerous through being infected by a virus. New to the idea of cancer viruses, 8 was that nucleic acid, when in the form of RNA, could be converted into DNA by the enzyme, thus providing a mechanism for viral nucleic acid to be inserted anywhere in the chromosome of the cells. 9. These new viruses became known as retroviruses. 10. The insertion of certain retroviral genes was thought to trigger cancer. The idea that these postulated viruses caused cancer quickly became hot news the world over, but did not survive investigation. 11. And other explanations were sought. 12. The theory did not predict or explain the dramatic increase in cancer cases. Cancer could not be shown to be transmissible, nor could it suggest any remedy in the form of a vaccine. 13. Interestingly, the spread of cancer viruses was blamed on homosexuals, prostitutes and black people, just as AIDS came to be 13 years later. 14. Whenever and wherever reverse transcriptase activity was detected, it was rashly assumed that retroviruses were at work. This turned out to be a grave error, because it was later found that the enzyme occurred in all living matter, proving that reverse transcriptase activity had nothing to do with retroviruses per se. 15. Repetitive elements. Further research showed that at least 10% of mammalian DNA was composed of repetitive sequences which were referred to as nonsense genes, parts of which nonetheless were described as retroviral genes. They exist in their hundreds if not thousands. Some of them can even replicate independently, and jump within and between chromosomes, and for this reason became known as retrotransposons. In the laboratory they can be made to migrate, and when this happens reverse transcriptase is invariably detected, which underlines the fact that reverse transcriptase activity has nothing to do with retroviruses as such. 16. Lab, HTLV3, HIV and all that. Because all this was already well known in 1983, it is incomprehensible that François Barsanussi, a member of Montagnet's group as well as Gallo's group itself in 1984, claimed to have discovered a new virus, when all they did was to demonstrate reverse transcriptase activity and to publish photographs of cellular particles without proof that they were viruses. They could neither isolate them nor show that they were responsible for creating the observed reverse transcriptase activity nor the tissue abnormalities from which they were obtained. 17. They concluded the role of the virus in the etiology of AIDS remains to be determined. 18. What makes a virus new? The isolation and purification of a real virus is a straightforward matter, because unlike cells, viruses of one species are always of the same size and shape, and can be readily separated from other cell components by standard techniques. A control experiment is to try an isolation with putative non-infected material in exactly the same way as the supposedly infected material. Nothing should be isolated in this case. To identify a virus definitively, a first and simple step is to photograph isolated particles of it in an electron microscope, and they must look like the viral particles observed in cells, body fluids or cell cultures to distinguish them from other cellular particles which look like viruses, but are not. Proteins making up the viral coat must then be separated from each other and photographed. This produces a pattern which is characteristic of the species of virus. A similar separation and identification procedure must be gone through for the DNA or RNA of the virus. Only after the viral proteins and nucleic acid components have been properly identified, is it legitimate to speak of a new virus. No evidence for the existence of HIV. Such evidence has up till now never been produced for HIV. No photograph of an isolated HIV particle has ever been published, nor of any of its proteins or nucleic acids. No control experiments as mentioned above have been published to date. What has been shown are photographs of virus-like particles in cell cultures, but none of isolated viruses, let alone of a structure within a human body having the shape ascribed to HIV. What the whole world has seen are models representing HIV with dish areoles, said to be receptors with which the virus attaches itself to cells. The existence of HIV is inferred from an antibody test. But how this is supposed to work, when the virus has never been shown to exist and obtained free of contaminants, remains a mystery. The